So hi everyone, my name is Kiel Telesford.
I'm Rachel Sattler.
My name is Michael Carapezza.
And welcome to, I guess, the first or second Bioforce, New York Bioforce class and session. So I'm just gonna review briefly last week at Ziva, right?
Yes.
We did a couple things, basic wet lab skills, take them with most students, we play some wet lab groups, a couple of dry labs as well, right? So, a couple of computational labs?
Yeah, thanks.
Okay. Basic lab equipment, they're all sorts, they're very sensitive. The tools that you're going to use today and you learn how to use, they're kind of expensive, so be careful, you'll learn how to use them carefully. Lab etiquette, careful structured approach is best. You keep records, you want to be safe. You've had basic bio classes in the past. You keep a lab notebook, you're going to take careful notes so people can reproduce your work. And of course, safety guidelines, safety first. If you're concerned about anything, you have any questions, you ask, right? So for today, what's today gonna look like? We have this intro session, you're gonna get to know who we are, our stories, do a bit of, exercises to brainstorm and reflect, which is important. Do a bit of health and safety in the lab, do some actual hands-on stuff, make buffer solutions. We'll have a lunch session. After, we'll review what we've learned, do some quizzes, practical assessments, and then a closing presentation by Rachel. So, who are we? So Mike's gonna kick things off.
All right. Hi, everyone. As I said earlier, my name is Mike. I'm the program coordinator for HYPOTHEkids Lab, so some of you guys already know me, of course. But basically, I got into science in high school. My high school was not academically rigorous at all, but I ran cross-country, and my cross-country coach, who was my mentor, was also a biology teacher and the head of the science department. So I kind of got into it through him, learned about what biomedical engineering was, wanted to go into it, so when I got to college, I decided to explore that. And for me, exploring that was similar to what a lot of you guys are going to be doing this summer. I basically decided, okay, I'm just going to work in different labs and see what it's like, all the different corners of biomedical research and biomedical engineering. So when I was an undergrad, I actually worked in 3 different labs, one that did cartilage tissue engineering, one that did lung tissue engineering, so that was really cool when they were trying to grow artificial lungs from stem cells in a bioreactor, which is basically a giant machine that keeps cells alive and going. And then I actually worked in the brain imaging lab where we did EEG and fMRI at the same time on people while they were doing attentional tasks. Often I was the subject also. That happens when you're the undergrad in a lab like that. And so for me, it was really about exploring different options, trying as different types of research as I could to see which one I was interested in. And so these are some diagrams from the 3 labs I worked in, just to show you how different, even in biology or biomedical engineering, how different the things you do can end up being. Right, so I mentioned I did brain imaging, right? That was basically putting these EEG electrodes on someone's scalp and having them do a task, right? Watch a little circle and click a button when it turns into a square, things like that, because we were testing attention, your ability to pay attention in boring tasks over long times, and what that would, basically what you could see in somebody's brain patterns while they were doing that. Up top you see something that's a little more wet lab, a little more close to what you guys will be learning about today in the lab. That was the lab I mentioned I worked in where we were doing cartilage tissue engineering. Does anybody know what tissue engineering is, or have a guess what that might be? Michael, yeah?
Isn't it taking like certain cells from a person in a way like regenerating it or attacking their body?
Right, you can take cells from a person, an animal, and then yeah, like he said, growing them in the lab, regenerating them, sometimes guiding them to become a different type of cell using different chemical signals. And you can do that to build up these cellular or tissue constructs in the lab, right? Little, basically objects you build out of cells, which you can theoretically then place back into somebody's body to repair damaged tissue. So in that lab, we were working on cartilage. The idea was you create these little, like, sort of cylinders of grown cartilage, and then implant them, in that case, into dogs or pigs that had arthritic legs, to basically see if that would improve their mobility and things like that. And then the last lab you see was also tissue engineering, and I mentioned it was lungs. That one was a bit different. We were actually taking lungs and decellularizing them. Does anyone want to guess what decellularizing means? It's kind of in the name, but anyone? No. Yeah. Yeah, you take the cells out of tissue. It's really small, it's kind of hard to see, but you'd be surprised at how much an organ still exists when you take all the cells out of it. There's a lot of non-cellular material in your body called extracellular matrix and things like that It's the stuff between cells. It holds everything together. And so what we were doing was basically taking all the cells out, leaving that extracellular matrix, which is basically the structure of the organ, and then seeing if we could repopulate it with new cells that we grew in the lab from stem cells. Because we were focused on lungs, the idea was, if anybody's heard of COPD, does anybody know what COPD is? Chronic obstructive pulmonary disease. Basically something that if you are a lifelong smoker, you can end up having this lung damage. And so the idea was to see if we could grow new lungs in the lab with fresh cells made from stem cells that wouldn't have those problems that somebody with COPD has in their lungs to improve their ability to breathe. So after I graduated, the last research job I had was a lab tech in a lab uptown at the medical center. Dr. Lisa Olson, she studies hearing. And basically what we were doing was studying everything about the hearing system, the auditory system. I was the lab tech. Does anybody know what a lab tech is or what they do? Yeah, so lab tech basically just like helps everyone in the lab do their research. There were 2 PhD students, a postdoc, and a master's student. They all had different experiments, so I'd help them set up, run their experiments, do data analysis. I also had my own little project where I was studying the tympanic membrane, also known as the eardrum, which you can see here. And we were specifically trying to understand how sound gets across the eardrum into the cochlea, which is the part of your ear that actually turns sound into a neurological signal that goes to your brain. So you can see that better here. Here's your eardrum, right? Sound comes in, it vibrates that eardrum just like a drum head. And then you actually see those blue things here. These are bones, or they're called ossicles in the ear. And basically what happens is the eardrum actually vibrates, moves those bones, which end up moving fluid inside your cochlea, this little like snail shell spiral organ in your ear. And inside that snail shell spiral, these waves would move and they would turn neurons on basically and have signals start going to your brain to say this is the sound you're hearing. And so we were interested in understanding basically how that drum head, that eardrum, could move in such a way that it was perfectly transmitting sound to your ear, to your cochlea. So we did that with a bunch of different experiments, did a lot of animal experiments on gerbils, to basically try to understand what was going on in that process. But that was my last lab job. I kind of moved away from research science, unlike our other 2 instructors. I kind of went more into like education stuff. After that lab, I worked at the World Science Festival, running their digital education website. And then about a year and a half ago, I ended up here helping Dr. Kyle run the HK Maker Lab program, which, funny enough, is still also research science. So now I don't study like biology or these mechanisms inside the body, but actually study how programs like this and programs like our other program basically improve or help people discover STEM, figure out what they want to do, maybe just get more interested in it or learn it better. And so now I do research of that more social nature through education. But still doing research, still use a lot of the same tools and that you guys are going to learn about today, not necessarily the physical tools in the lab, but more of the conceptual tools of how to think like a scientist, how to ask questions, and how to start figuring out the answer to those questions. So I think that's something, as I wrap up and pass on to the next instructor, keep that in mind that even if you end up working in a lab that maybe doesn't do a ton of the stuff you do today, a lot of what you learn about how to think like a scientist, how to be like a scientist, is transferable to almost any type of research. So you can make big jumps in what you're working on and still use these same fundamental scientific skills. Okay, so that's my bio. Rachel's up next.
I'm very excited to be here. When I was at Columbia, I TA'd for the HK Maker Lab for a couple of years, and I had some of the HK Maker students work with me in my lab, the Bone Bioengineering Lab at Columbia, and it's a great experience. I'm really excited for you guys to get all this exposure. My path is crazy to science. So when I was your guys' age, I was a very serious ballet dancer, and I graduated high school and actually joined a ballet company. So I danced with the Sacramento Ballet for a while, and then I moved to France and I did contemporary dance, and then I moved to New York and I danced for like a decade, and then I went back to school. So now I'm in a science career. I work as a research associate at Regeneron Pharmaceuticals up in Tarrytown, and I work in the group called the Viral Vector Technologies Group, and we do something totally sci-fi sounding. I engineer viruses, I reprogram viruses, and if you don't know, viruses essentially are really good. They attach to a cell and they inject their genetic information into a cell, and they take over the cell and they make the cell basically replicate their genetic information. So we are trying to use that, because viruses are really good at getting genes into cells, to put in the genes that we want. So genes, like if somebody's missing something, or we can deliver some type of therapy. So I re-engineer viruses to deliver a therapy instead of a disease, which is totally wild. But I started at your age as a ballet dancer. So I want to let you guys know, like, I think at this age, when you're getting ready to graduate high school, you feel like, all the adults, they all know what they're gonna do, and like, you're supposed to know your path. Don't worry, it's like, you'll figure it out as you go. So this is me in 2012. I had started school at this point, I'm the one in the air. But just an idea of what I did, like science-wise, so I went back to school. I went to Fordham University because they had a great program actually for adults returning to school. And I ended up doing my bachelor's in engineering physics. I went back because dancers wear out their hips, as you can imagine if I was doing that with my hips since I was a little girl. So a lot of dancers get hip replacements in their 30s. My best friend, who's 37, already has a hip replacement. So I thought, I want to go design hip joints, and I can make better hip joints. So that's how I ended up in engineering physics. But then all my intern experiences and like undergrad research experiences kind of shaped the type of science I'm in now. So obviously I'm not designing hip joints, I'm engineering viruses. So my internships I had, and they were all sort of non-traditional as well. I did one with the engineering physics department. That was the first experience I had. That led to a summer experience at Sloan Kettering Cancer Center, where I got to work in a molecular biology lab. And then I went to a conference, like a seminar locally, and I saw this woman who was doing really cool work with tendons. So I basically emailed her to see if she could use help in the lab, and then I worked for about 6 months in that lab my senior year. And I'll tell you guys a little bit about those projects. And all of that ended up, I was here at Columbia. I did a master's in biomedical engineering and part of a PhD in the bone bioengineering lab. And I ended up, that path wasn't quite the path for me, so I started at Regeneron about in November. So I've been there 6 months. But I just wanted to give you an idea, like Mike did, of the projects I did when I was an undergrad and like young in my science career. Maybe not in age, but in my science career. Because you'll be in your internships working on a little part of the lab, but you get to learn so much about all these really interesting topics, and the more experiences you have, it helps you understand what you're really interested in, and it will help guide you as you're choosing your classes in college, or what your path might be afterwards. So the very first experience I had is up in this top left corner. This was in the engineering physics lab, and I basically assembled that crazy laser with all that equipment. I didn't really know much about lasers at the time, but I definitely learned. And so essentially this whole setup, this is a quantum cascade laser right here, it's a little tiny part. And the idea of this project was that we would shoot this laser through, actually let me back up. We had melanoma tumors, and tumors let off gas as they're tumorizing. As they're like, so we wanted to see if we could essentially smell the tumor with the laser. So we would put the tumor in a tube, and then we'd shoot the laser through the tube and detect the laser at the other end. And if there was a shift in our reading, that meant that the laser was being absorbed by something in the gas, and that could help us figure out what that signature was and maybe design a tool that would be able to smell the tumors. And actually it's really interesting because dogs can smell certain types of like lung cancer. So they actually do tests where they have dogs smell breath of people with CPOD or lung cancer, and they can actually correctly identify pretty accurately whether or not a person actually has that disease. So our jobs might all be taken over by dogs someday. But so the whole summer was basically like building this and getting all this equipment to work together, which was hard, but I learned a lot. And then there was a computer program, and I used this program called LabVIEW, and I had to make adjustments to it, so I got to do some programming too, which was awesome, and I really liked it, but I like biology more, so I wanted my next experience to have more biology in it. I was from dance, I was really interested in the human body. And the tumors we were getting were from this lab at Sloan Kettering. So again, it's sort of like through my mentor at Fordham and talking with the person we were collecting the tumors. It wasn't a traditional program even. I ended up in this summer position at Sloan Kettering in the White Lab, and they worked with zebrafish, which are these tiny little minnows. And one of the things they had done is they had genetically modified these fish so that they would be clear, so that they could study how the tumor metastasizes, or how the tumor gets from where it's one fixed location and then spreads through a body. So here they would inject the tumor into this clear fish, and then over time they could see how it metastasized and where those tumors went to. And so my actual role in that, I mated zebrafish, so I got really good at telling male zebrafish from female zebrafish, which is not easy. There was a special device called the iSpawn that actually my PI in the lab, the PI is the principal investigator, they like head up the lab, helped design. And we would separate the males and the females because we wanted all of the, I was studying zebrafish embryos for my project, so I wanted all the embryos at the exact same age. So at night, I would put all of the female fish in the bottom of the iSpawn, and then they were separated by a net, all the male fish on top. So I'd have to tell which was which and use nets. And then in the morning, I would basically dump the male fish onto the female fish, so it would like rain men. And they would go crazy, and then they would lay all these eggs and fertilize them and drop them into a funnel, and then I would collect all the eggs and do my, hatch the embryos and do my experiments. So you never know what you're gonna end up doing in your lab. But I learned some great skills, I learned what you have to do to get into an animal room and a lot of that as well. And about timing experience. And then what I was doing is I would take these embryos and I did this procedure called an in situ hybridization, which is basically I would treat the embryos with different chemicals, and then I was using a probe that would stain purple on a particular type of cell, and I was looking to see what chemicals made that cell more or less. And it's a cell that we know if it gets modified at the embryo stage, if there's a mutation, it can lead to melanoma. So I was doing this little project, and a lot of it was just trying, again, to optimize this project. That's a lot what you do when you're an intern. You're often working on something that the grad students don't have time to figure out, and you're helping them figuring out. And actually, a person that's just getting ready to defend in this lab, 4 or 5 years later, this ended up being part of her thesis. So that work helped her, me figuring this out, helped her move on in that project. And then the other lab, I told you I went to this seminar and I saw this really cool talk about tendons, and I knew about tendons from dance. So I ended up working at this lab, the Andrew Ross Curry lab at Mount Sinai in the orthopedic research department. And they worked with mice, and they had this really cool mouse called the MRL mouse. The MRL mouse is like a Wolverine mouse. It heals without scars. And that's obviously very unusual, so we want to study why is that happening, because if we could figure that out, we could maybe figure out how we could heal without scars, which is very important. When you work with mice, a way that they identify them so they know which mouse got which drug or injection, so you do a thing called an ear punch. And so you might have one mouse with the right ear punch, and one with the left, and one with 2 on the right, and that's how you tell who's who. And what was happening was there's a particular strain, a B6 is like a wild type mouse, it's just a brown mouse, loud mouse. And there was a particular strain of mouse that some poor grad student was ear punching, and then a month later there were no ear punches. They didn't know which mouse was which. And I imagine they were getting in big trouble all the time from their PI, their boss, for not doing this properly. But it turns out that this mouse, and a lot of this tissue can heal without a scar. So what our lab wanted to know is you have a muscle in your shoulder, several muscles called the rotator cuff, and this injury heals horribly. So there's a lot of people that even with surgery it never heals. So they wanted to know, does this heal on this mouse, and can we figure out what's different about how it's healing so we can maybe give people a drug that's going to help them heal or understand that. So I didn't get to do these surgeries because I wasn't mouse certified at that point. But there was a surgeon that would come in. He was a peds surgeon, so he worked on babies, and he would do plastic surgery on babies, if they had a cleft palate where this doesn't come together. So it required really fine skills, microsurgeries. So who better to do a tiny microsurgery on a mouse shoulder, right? So the surgeon would come in and cut the rotator cuff and then repair it for us. And we would use our Weil's type mice and our MRL mice. And what I would do is take this tissue and stain it. It wasn't so different from this actually. And I would look at the types of cells that were in that tissue that was healing in our Wolverine mouse versus our Weil's type mouse. And so I learned tons of skills there, and they're actually skills that I brought to the BBL when I did my my graduate work, and now in my job in viral vector technologies, the skill I learned here it's actually a very sort of complicated, tough skill to learn, and I actually brought that to my lab now. So in a big company, I added value from something I learned in essentially an internship as an undergrad. So, but all of this, like, you know, I knew about dance going into all of this. So you guys will be amazed when you come out of your summer internships. You will have little projects, and you will know a lot about one thing, but you will learn, and it's really exciting. And I think what we want to get across in this whole course is that we all started making buffers, like what you guys are going to be doing over these courses and learning what a pipette is and learning this terminology. And now we can talk through much more complicated things, but it's totally within all of your grasp. So I think that's all for my intro. Let me go to Todd.
Thanks, Rachel and Mike. So, yeah, my name is Kiel Telesford. This here is a map of the Caribbean. This is where I'm from. I was born in Trinidad and Tobago, but moved to New York City. I was about 5, so grew up in New York City. Went to high school for environmental studies, around 57th Street near Beacon and LaGuardia. And I actually also did a program similar to this at Columbia, but on 168th Street called the CURE program when I was in high school. So, like I said, and like Rachel said, you know, we were where you are, and you can go all sorts of places, right? Fun factoids, like the Yankees and Knicks, or Arsenal and SCFC, you know what those teams are, fans of those things. Not a fan of gentrification, but that's a talk for another day. So my regret for this slide is that it has a straight line, linear sort of thing. Of course, hindsight's 20/20, and it's definitely much more like Rachel's thing, where you're like not sure what you're doing. You think everybody who was ahead of you had it all figured out when they were at your stage, and definitely not the case. So don't let the straight arrow sort of fool you. So yeah, I started high school, of course, right, did the CURE program. And at that point, like most, first 1.5 second duration immigrants, was like, all right, medical school, gotta go to MD, right? Your parents want you to do it, make that money, get the respect, all that kind of stuff. There's nothing else in life, just medical school, right? Well, obviously, you all know, you know where I come from. Went to Oberlin College. Our mascot is the albino squirrel in Ohio. Did my bachelor's. First heard about the PhD, so what is that? Did a bunch of undergraduate research experiences all over the place, at Oberlin, at Virginia Commonwealth University, at Duke. And again, that's helpful for perspective, which is going to be another important point that we're emphasizing. This experience gives you perspective, and you can carry that mentality all the way throughout wherever life takes you. Continue to build perspective. So the undergrad research programs really helped give me an idea of what was out there and what I might be interested in. I followed that up in a post-baccalaureate program. You should make a note of that as well. These programs that you do after undergrad that can further help cement your interests or give you more experience. And it was there that I realized, you know what, MDs, physicians, even though they're like in the limelight, what they really do is manage patient care. What I want to do is actually cure disease, right? And in speaking with these MDs, while throughout this experience I was shadowing all that kind of stuff, there was this undercurrent of frustration with the medical system, with their lifestyle, and even that limitation, right? They're like, I can only do so much for my patients. I have so much paperwork to do. I barely have a life. I'm just managing symptoms. So you actually do have a lot of MDs that try to go into research as well. You can do. But nonetheless, at that point, I started to realize, you know what, the research thing is actually really important. That was further cemented. Yes, even while I was in graduate school, getting my PhD, I was still thinking about med school afterward, right? Such as the immigrant sort of drive mentality, right? And I was seeing firsthand all my med school friends living in the library, memorizing facts. You get tons of debt after med school, constant pressure throughout the training and afterwards. I've heard described as a second marriage once you're a physician. It's like, you know what, I just want to get married once. So all I have to say, I ultimately decided to continue with the research track, decided to forego the med school track. For this reason, primarily, the highlighted reason, I want to better understand and treat disease through research. So I'm currently doing a postdoctoral fellowship at Cornell. My PhD is in immunology, so I learned about the immune system and studied immunology. So as I mentioned, I'm a biomedical researcher. I'm doing a postdoctoral fellowship at Cornell. I currently study a disease called multiple sclerosis. To better understand how it works, so we can treat it more effectively. That's really kind of how this whole thing works. To better understand, disease or whatever mechanism, whatever pathway or cell type or model organism, better understand certain mechanisms, certain biology, usually in order to understand certain disease, so we can treat it, something like that. Okay, so what is MS? Multiple, several, many sclerosis refers to scars or plaques or lesions that are found in the central nervous system, so the brain and spinal cord. You imagine you use your brain and spinal cord to do all sorts of things, right? So to think, cognitive function, to move around, handles other sort of functions, let's say, in your body. So if you have these scars on your brain or spinal cord, you can have things like paralysis, cognitive dysfunction, and other sort of bodily issues. So it can be a devastating disease. This is a cross-section of a post-mortem brain, human brain, and the arrows point out these discolored plaques. So that's kind of what they look like in a preserved brain, of course. It's been fixed. In real life, when you're seeing patients, you have magnetic resonance imaging, MRI, to really view what this looks like when a person is alive. So it's the cervical spinal cord, and their arrows pointing out these slight discolorations. They use a contrast agent called gadolinium to highlight these lesions. So there are a few on the spinal cord there and a few throughout the brain. And that's what it looks like. And so, that's what MS is, literally. It's a name, the term. As far as, we don't know what causes MS, but we do know that what drives part of the pathology is this recurring damage that's facilitated by white blood cells. So for some context, your central nervous system, again, your brain and your spinal cord, not to be confused with the peripheral nervous system. So just your brain and spinal cord. And within your brain and spinal cord, you have a bunch of different cell types. You have neurons, you probably heard of those. You have this other type of cell called glia. And these cells all function together to basically allow you to function, to think. We don't know how that works. Still working on that part. So nonetheless, you have your CNS, you have white blood cells, your immune system, part of your immune system that's all throughout your body, in your tissues, in your bloodstream. And these white blood cells, unfortunately, through a sort of autoimmune process, enter the central nervous system and promote these plaques. So as far as the approach that we're doing in the lab, just very briefly on some of the tools. So the sort of general question, as you probably learned in basic biology class, and as you see as we go through scientific method, starts with some kind of question, an observation, a general question. And this is a very, very general question, this is a general approach. So which inflammatory mechanisms are overactive in patients with severe MS, those who have severe disease versus mild disease? And with that, compare various inflammatory responses in B cells, which is a type of white blood cell, to see whether or not we can delineate what makes that difference between the severe versus mild disease. So what does that look like? Literally, get blood samples from patients, isolate white blood cells, including B cells, from them. So we use different techniques to isolate the cells and then model the different types of inflammation and measure inflammatory biology. So these are just my general take-homes from our couple of slides. We're normal people, the three of us, mostly. Mike's from New Jersey, so
I'm definitely a bird.
Okay, so maybe not so normal, but as normal as you guys are. So we're driven by our interests, ideals, solve biomedical problems, scientific inquiry, communicate messages. You guys can do all this stuff too, right? You're just starting out. You started where you are, and this kind of a little bit. And also, again, I want to highlight life experiences, such as near Bioforce. Take advantage of them, gain perspective, take time to reflect, to know yourself. And that's a continual process that happens all throughout. And by doing that, you can more effectively sort of navigate that topsy-turvy path and make decisions effectively. So that's it for the introductions, our introductions. It's more like a lecture classroom thing. We want to be a bit more interactive and some guys on it. So we incorporate a couple of just reflective activities. It's good you guys have sheets of paper. So right now I'm gonna take 5 to 10 minutes, reflect on these questions and take about a couple minutes to do it on your own, write something down, then share it to your neighbor and share it. So why are you participating in this program? Be vulnerable, be open. It could be that your parents just wanted you to. They're like, look, do this thing. It's at Columbia, it's an Ivy League school. It would go down your resume. Write that down. Are you actually interested in science? What are you interested in? And what do you hope to gain from it? And you can talk as well. So who wants to share? Start away from what you got.
Well, I joined the program because I'm going to major in biomedical engineering in college this coming fall. And then I saw that this program was biomedical engineering, so I was like, I might as well get like a head start and see what I enjoy and what I don't enjoy.
Cool.
That's great.
What's your name?
Caleb.
Kiel.
It's really broad. There's a lot of things you can do in that topic, so it's nice. And you'll get exposed to a lot in this program. It's great.
Hi, I'm Marsha, and I'm participating in this program because I like science, and my school doesn't really have like a formal lab or anything, so it's just learning and then preparing for the test, the regions. So I really want to get like hands-on experience rather than just study for a test.
Okay, so I've been really interested in science since I was younger, and I thought that I really wanted to be a science journalist, like research about science and write about it and publish it, because I really like writing too. But then I also realized that I kind of like looking at a problem and trying to solve it through science and engineering. So when I did the HYPOTHEkids lab over the summer and I got to experience that, I realized that I kind of wanted to do something like that. So when I do this 5-year Bioforce and do the internship, I really hope that I can find that sort of thing.
Yeah, definitely sort of.
What was your name?
It's
And Genevieve will be an instructor joining on the 2nd. She's actually got into career in scientific writing from her PhD, so she'll have some good insight into that role.
You guys are thinking in the right way. New brand new experiences. It's fine. You all start at some point. Preempt those points ahead of time, and you don't know until you tried it. Right? So you have to explore. Okay, so as was probably outlined the week before, these next couple of weeks are sort of prep for the summer. You guys are going into professional biomedical research labs. And so the objectives for these next couple of weeks are as follows. Really literally sort of what could happen in the summer? What to expect, right? Probably a lot of questions for a lot of you. It's probably your first time entering into an environment like this. What's it like to work in an academic research lab? What am I literally going to be doing? Am I even ready for this? I feel like I'm in high school, people are PhDs and MDs and all these other degrees. A bit of this preview. It'll be okay. You'll be fine. We'll get into that later. Practical skills that was mentioned by Rachel. So later today we'll make some solutions. You guys already started to learn how to pipette. I think last week. No solutions last week.
Yeah.
Okay, solutions. So more solutions, pipetting. And throughout the next couple of weeks, we'll learn some basic tools, some basic techniques that are used, frankly, in many different types of research. And the aim here really is familiarity. So we're not saying like, oh, but in these couple of weeks you're going to be experts and you're going to just jump in the lab and do anything. It's not just familiarity, basic tools. And finally, and arguably, and most importantly, is to develop, what's what we refer to as a problem solver mindset. Kind of alluded to it, in that, having gone to high school in New York City as well, and many other high schools across the country as well, right? You kind of learn, it's like, all right, here's your material, read it, here's some problem sets, you kind of regurgitate what you learned, the little facts, and typically that's it. But when you're dealing with complex problems facing the world, problems facing society, disease, and that kind of thing, there's no answer key. Right? So it's more problem solving, that kind of mindset than all right, let me just memorize this stuff. Or somebody knows the answer, just gotta ask the right person. So those are the three objectives.
And just to add to that, I think that's a real strength of biomedical engineering, is you learn to solve problems first, and that's the goal right at the start. Where not like other science modalities are fantastic as well, but as you alluded to, you have to learn that one problem and all the facts around it. And in BME, it's often the focus is on a general, how do I solve problems? And that makes you an excellent researcher, and you can transfer that to then whatever disease you want to study.
Actually, I appreciate that one, because earlier today, I was chatting with Mike, I was like, the thing I appreciate about BME people is that you guys really have the best of both worlds in a sense. You have this, for those who are in the HYPOTHEkids lab thing, this idea of design thinking and ideation and coming up with prototypes and that sort of thing. Then you just supplement that mindset with the knowledge of a certain field. So it's not a bad choice. Okay, so just a few slides on how academic biomedical research labs work generally. Starting with who's who and what do they do. So Mike sort of alluded to a couple of these positions. Who are these people? What do they mean? So each lab's going to have a principal investigator, or PI for short. This person is the most experienced. It's basically their lab. So the Telesford lab. Not yet. But whatever lab. They're the boss. They're the person that sets the scope and trajectory for the lab. They primarily write grants and fundraise, so depending on the size of the lab, they might also jump in and do experiments too. So that's not very common. This person you'll at least meet, you'll probably interact with a little bit, but you're not going to necessarily look to them for a day-to-day interaction and guidance. Next, you have postdoctoral fellows, such as myself. We're sort of like, PIs in training, if you will, right? And so they train us we get more independence than we had in grad school. We usually have more responsibility. We're involved in several projects, not just our thesis project. Primarily day-to-day, these people, myself, we read articles, we design experiments, we execute experiments, we also help manage the lab. Chances are, a number of you will probably have a postdoctoral fellow as your primary contact during the summer to provide you with guidance on your own, on the project that you're attached to. Then you'll have students, as Mike also mentioned, so PhD students, master's students that are in the lab to learn the craft from the postdoc and from the PI. They're also doing experimental work that focus on a thesis project primarily, reading articles, they'll sometimes be taking classes. You may not be, paired with the postdoc, you might be paired with the grad student, PhD student, or a master's student who's in the lab. And likewise, they're equipped, and probably in a sense even more so, to just meet you where you are and help you learn the techniques and guide you through the process. You'll also find in some labs, undergraduates, even high school students such as yourself, though high school students are less common, so you guys are a special group. And so unlike grad student postdocs, you'll be attached to a project that's going on in the lab, but primarily in this, they know that you're not learning this stuff, so don't put pressure on yourself. Oh my gosh, you're going to make me. You're going to think that I need to know all this stuff. I don't know any of this stuff. They know. So you're primarily going to assist in experimental work. You're not necessarily going to design experiments per se, though you may help. They will guide you in that. So it's a guided process. You'll be doing actual techniques and assays. What that word is. But even if you're not assigned to read articles and learn about the project, we'll emphasize that you do so. Why? Because that's honestly where the bread and butter of this whole learning takes place. The techniques, even though they look impressive, and it's like, oh, my lab coat, I'm doing this stuff in this fancy machine. That's just process in a sense. I mean, there's some skill involved, but that's not where the creativity actually comes in per se. It's the stuff that's done behind the scenes, the question that's asked, the experiment that's designed. So don't get caught up in, I learned how to do gels and do this and that. Great. What was the story? What was the question that was asked? What's the problem that's they're trying to solve? being able to explain that is perhaps the most important takeaway I would say from this experience.
I
Do you guys say
just
so?
want to add to that too. In a lot of these labs, the questions they're asking and the story they're working on, it's not even published. So like it's not common knowledge, and you know, maybe no one knows at the lab what they're discovering. So you have a chance to really, if you ask questions and participate in that way, to learn something that very few people and actually the whole world might know yet. So like don't put in your head, I just need to go and learn how to pipette perfectly. Like you're in there to like absorb all this interesting and exciting information too, and you'll get exposed to technologies and experiments that are being invented like in front of you. And even if you're not directly in that, the other thing is scientists love to talk about their projects because they so rarely get to do it in the real world. So even you can ask not your grad student, like you can ask other grad students and people like what they're working on when you have extra time, and learn so much this summer.
Good. So a bit more on that, what's it going to be like. So labs vary in size and culture. Some labs will be huge, they make up buildings or several floors. Other labs are like 3 people, be a PI, a grad student, and maybe you. So I'm not sure which labs you guys are in, which institution even, but it can range a lot. Some labs can be really, really laid back, and it's like, oh, you guys are doing the work. Or they can be very, very intense, but it'll be okay. So they're usually not 9 to 5s. People usually come in like, oh, it's 9 o'clock, gotta get to work, and it's 5 o'clock, I gotta check out. It's basically, it's flexible. You go in when you need to go in. Well, they do anyway. I'm not sure what the requirement is. Students. But the key there is that it's flexible. It's not a 9 to 5 job.
Yeah, some experiments take 10 hours to
Yeah,
get through.
you'll have time points. You'll have to.
Yeah. I mean, you will probably have a more regular schedule
Yes.
as a summer student, but just know it's not a normal job in that
Yeah.
sense.
I think my craziest time was when I was recruiting patient subjects for my study, and that's the thing with when you're doing with humans, and, they'll have an appointment, they'll be late, all this stuff happens, you end up getting sample hours later than what you expected. I finish at like 3, 4 in the morning, then head home, sleep for a couple hours, come back in and do the same thing again. That's not typical, but that can happen.
Yeah, and cells and mice, the things that you're using for your experiments, they don't work 9 to 5. So sometimes you go in and your cells haven't grown to the right place you need them, because they don't have a clock in that incubator. So it's definitely, you have to be flexible working in a lab, and if you want to go into a job in science.
Right, so as I mentioned, you're going to be assigned to a project and a day-to-day sort of contact person. And again, I can't stress enough, there's not going to be any real pressure for you to come in like knowing. So they probably will be pleasantly surprised. They're like, oh wow, you guys know how to pipette, you have an idea of like how to calculate this thing. That's great. So that gives you the freedom to learn, right? The pressure's off. Make mistakes, have fun, right? Ask questions. Okay, so how do you make the most of this experience? You get what you put into it. It's not gonna. Don't just sit back. You can sit back. You can show up, and be very casual about it, but that's all you're gonna get from it. And if anything, you're less likely to get a strong letter of recommendation if you ever ask them for it.
Yeah.
So it's an opportunity to practice bold, purposeful inquiry, ask lots of questions, be proactive. Initiative is always looked upon with delight from researchers. Be teachable. So I mentioned the lab work, the experiment stuff is interesting, it's fun, but that's not the core of it. Understand the scope of the project. I put ask questions, but with that proactivity piece, it's also about we don't know everything. Despite all the schools, the PI is not going to know everything. The postdoc's not going to know everything. The grads, we know like this narrow section of the stuff we've been working on. We don't even know I don't know all of immunology. Again, I'm not like this. That's not how it works, really. So we're still all human. Chances are you're going to come with there will be times you'll go to your postdoc or grad student data person and say, what does this mean? Or how do I do this? And I'll be like, I'm not sure. I'll get back to you. Or let's look this up together. So,
Or,
yeah.
I think the best compliment you can get is when someone says, "That's a great question."
Exactly. That's a common one.
Yeah, and that's great. And those are the types of things that go into those letters of recommendation that your mentor ends up writing for you when you're applying for the next thing. Like, so and so asked, great questions, and they were always very curious and engaged. It's not they're amazing at doing gels. So it's that you can start to think like a scientist. But I've definitely had the experience too of explaining an experiment to a student that was a high schooler, and them asking me, wait a minute, you said, you need to control things, and like, don't you need this type of control? And I was like, actually, you're absolutely right. Because we get all caught up in our own projects that we're working on, and we miss sometimes the forest for the trees as well. So it can be great to have somebody that's newer that remembers those basic things that sometimes you forget when you get really technical.
Right. Actually, in case you guys were taking, just put this up there to kind of like signal a little bit of a break, but I think you know what's coming. There's actually a website called PhDcomics.com, and these screens are kind of small, but it kind of shows the same hierarchy of the lab. So you have the PI faculty, then you have the postdocs, so that'd be me, so like, oh, I see the PI, right? Then, in the basement somewhere, you have the PhD student just like working away, master's student huddled in the corner, just like stressed out, and then you have the undergrads, and I guess the high schoolers too. So don't take that personally. It's just a comic. Okay, so in closing, life is beautiful. The world's a wonderful place. I'm not sure where Wakanda was, but I just put it on. It's an amazing place. A lot of stuff. It's also complicated, right? And we have all these social problems and political problems, and economic system, and environment, and healthcare, and all that kind of stuff, right? So it's beautiful. It's complicated. It has a lot of problems. The main problem is that there's no answer key, right? So this is what we do. This is what we want you to sort of generate and start to go in that direction as far as being problem solvers. And so we make progress on those problems for which there are no concrete solutions that any one particular person knows through this inquiry process, through the scientific method. So what's the right answer? Nobody knows. How do you find out? The answer is over time, with critical thinking, and with collaboration, as we'll also go into a bit later. So usually, just taking a step back in biology class or AP Bio or whatever else, there's like you get to go into a lab and you have or your project, it's like this worksheet, it's like ask a question, generate a hypothesis, write your methods, and do experiments and write a conclusion. It's like a nice, neat little package. Just there's an answer, and so you do it, and you get an A+ or whatever, and it's done. But it's not quite like that. But generally, this is how I tend to think of the method itself. There's a big problem, big complicated disease or whatever else. You take that big complicated problem, you break it down to smaller observations. So we know the immune system is involved and important for MS. So if I want to try and tackle and understand how this has worked, I think I'm going to start there. It's an accessible compartment. I can get blood draws from people. I can isolate immune cell types. I can do functional assays, functional experiments on those cells. With those small observations, I ask questions that lead to testable hypotheses. Is antibody production by B cells in this particular cohort of patients linked to lesion load and volume in the MRI and looking back at their medical records? So big problem, small observations, asking questions, forming testable hypotheses, reading the literature. You mentioned reading and observation, right? So people have probably asked similar questions, maybe even the same question. That problem might have already been there might be solutions out there, in a sense, to parts of that question. So I'll look at the literature. What have other people done? What are the tools are out there? Maybe the types of tools that I have can solve a part of it, but not all of it. So we mentioned biomedical engineering being really good at leveraging other fields. They're good at taking chemistry, biochemistry, cell biology, lots of different things to solve a particular problem. Try alternative approaches to validate findings. I might say, I got this answer using this approach. Let me see if it also works with this other approach that will give me confidence that my first approach makes sense and might be correct. And finally, collaboration, share your findings. Can other people repeat it? Which can be a problem, but that's what it is. And so the final sort of thing, another activity is wanted to do, reflect again, what sorts of topics you're curious about? What are your interests? What sorts of problems are you interested in solving? And this is not your final answers. It's like right now, it could be as broad, specific as you want, and that may change. It likely will change over the course of your education. But it's always good to reflect. So you want to give you an opportunity to do that now for the next 10 minutes. Just go for it.
Can we get a couple people to share their answers? Or yeah. Just make sure you say your name.
I'm Anika Wiki. So I did HK Maker last summer, and
Okay.
I actually became fascinated with bacteria and cells through. We would have presentations. People like yourself would come up and present. And I remember they were students at Columbia, and they formed teams just through how we did it, just doing random experiment. I think they were actually they had no idea what they were going into, and just through working out, they were working with bacteria and stuff. They were able to create their own company, and also another professor who was using everyday bacteria to engineering it actually cure cancer. And it's fascinating how such a small thing like bacteria is so complex, cells are so small,
Yes.
but they can do so much to cure.
Bacteria is
Yeah.
very interesting. And was it Tal? Do you know? Is that the lab they were in? Do you know? He's a newer professor working on bacteria.
I'll chime in and say yes, it was tough.
Yeah,
I remember that talk.
Because he works on really cool stuff
Yeah.
with the bacteria that glow and
And we also turned that into art, which was
yes,
really
that's
creative.
tough.
Yeah.
Yeah. Very cool, very cool. And I love what you said too. They also had no idea what they were doing. If you guys feel I have no idea what I'm going to be doing, that is totally normal, and you will continue to feel that way through your whole scientific career. Does someone else want to share something? Yes, and please say your name.
Right now I'm really fascinated with statistics because I expected them to be a lot more advanced today, but today there aren't really any advanced statistics where they can be controlled through the mind, and they're basically just attached to your body and use not through your brain.
Yeah, they're still very mechanical. Yeah. There's some really interesting challenges they have to solve still about energy, because you need to be able to power something, and you have to make the energy supply very small, so how do you make it last? And we still don't know so much about the brain, despite all these really smart brains working on it. Yeah, very interesting topic. And can we get one more? Yeah.
Yeah, so I'm Natalie,
Natalie.
and I'm really interested in neuroscience, and the main reason I am is because my grandmother had Alzheimer's, so I wasn't able to get that relationship with her the way that a lot of people have that relationship with their grandparents, but because of that, it was what how did this happen, how are people trying to solve it, because it's a very common problem. And so throughout I've just since then when I found out I was pretty young, I was what is going on with the brain here, why is it whenever what's going on with the neurons and everything, and why is the brain not functioning normally. So it's just since that time it's just and this is something I was really interested in.
Fantastic. Yeah, this is very interesting. And I hope over the course of this we get to find out more from everybody. But first, it's been a little over an hour, so let's stand up and kind of stretch out. If anyone needs to go to the bathroom, grab some water, and then we'll come back. And we have to do a little, I hate to say the boring stuff, but we have to do a little bit of health and safety, and then we'll just get ourselves on that. But let's take it's 11:14 right now. Let's come back at in 10 minutes. 11:25, but just a chance to go bathroom, just stretch your legs, and okay. So before we go into the lab, when you're in your lab, there'll be some type of requirement for health and safety. Keep you safe, so you know and you're aware of the hazards you're working with. It will be unique to the institution you're in, and depending on what you're working on, you might have to do additional trainings. Some of them will be in person and online. We're just going to go over some basics before we go to mixed chemicals today. Yeah, so this part's always the dry part, but we'll see if I can make a joke somewhere, I don't know. That will stretch my comedic abilities. But so like essentially the PI, again, that's the boss of the lab, like is really responsible and for making sure the lab's compliant. But you have to understand your roles, make sure you have training. Usually health and safety at institutions does do inspections from time to time, and they'll look for things like lab codes and are you pouring things you shouldn't be out of the hood and are things labeled, and the PI will get like notified if you're not compliant. So I got in trouble at work for not having safety glasses on the other day. They were on the top of my head, but not on my eyes. So know the rules of the lab you're in is the key there. And health and safety is usually super friendly and like desperate to have people be compliant. So like if you're ever unsure of how to dispose of something, or you might be asked if your mentor doesn't know, they're usually really happy to come do extra training or like help explain things to you. So the main things we want you to know today is there will be labels and signs all over the lab. They'll be on the chemicals. If there's radiation in the lab that's used in any of the experiments, or the old lab I worked in had a CT machine, you'll see those labels. There'll be lots of chemicals with skull and crossbones or big flames on them. You want to make sure you're handling things, like don't be scared of the chemicals, but make sure you're handling them properly. And you know, especially things that have like a skull and crossbone, where you should be using that and what you should do if you accidentally get it on you. So there's things that are a hazard, because like a physical hazard, like they're toxic, they can cause fire. There's also obviously like physical hazards, like if something is hot, don't stick your hand on it. Pay attention, lots of equipment has lights if it's on. So if something appears to be on, don't just touch it, make sure you understand, is that something that's a hot plate? Or, and obviously if there's spills in the lab, we're all responsible for keeping us safe. Don't assume it's someone else's responsibility, notify somebody if something's wet on the floor, there's something, like you can take responsibility to address that as well. And then there's also health effects from some of the chemicals. Again, you're not gonna be using anything totally crazy in this, but you just need to be aware of what they are and how to properly protect yourself. So if you are using a chemical with a skull and crossbones on it, or you'll see some labels, like make sure you know and you talk with your mentor and you understand, should I be wearing a mask? You always should be wearing gloves. Do I need to use this under the chemical fume hood? These are examples of some of the types of labels you'll see. So the physical hazards are on the top. Things can be flammable, like all the alcohols, things that end in an OL in the name, like methanol, ethanol, are flammables. They're usually stored in a particular place. When you're pouring those, you always have to pour those under a chemical fume hood. And you can never, usually, there's special, it needs to be cooled, there's usually special fridges that don't have any of the like wiring on the inside that those get stored in. There's things, there's compressed gas, there's things that can oxidize. Things that are corrosive can either eat through the table or eat through your skin. So you want to be extra cautious that you have your lab coat on, that you're using gloves, and that you protect the surface that you're working on. So that's the label with like the burning hands. So these are holes being burned through things. You'll see that on some of our chemicals. And then you've got the health hazards as well. So the big exclamation's a skin irritant. Some things that you don't think would be an irritant, when they're in a super concentrated form, they will be. And for the most part, these things, you wash. If you get it on you, you'll start to get red and you immediately go to the sink and you flush with water. And then you'll see things that are environmental hazards. Anything with this label never, ever goes down the sink. We don't want to kill all the local fish. So you'll be quizzed a little bit later on looking at some chemicals and just identifying the hazards. Just being aware, being observant. So on chemical labels, you usually have a product identifier. There'll be information about where that chemical came from. You'll have pictures that will show you what hazards on that chemical, and like potential statements. And there's often expiration dates. It's good lab practice anytime you get a new chemical into the lab to label that it's arrived. You'll see that on our chemicals today. And when you first open it, to label it when you open it. That helps. So a lot of labs have chemicals just stored on a shelf and they'll be there for months or years. And it's good to know when it was opened, because then when you suddenly have to use it again for an assay or an experiment, you can look up and check expiration dates. So these are just some other examples, and we can, I'm sure, Ziva, Mike, we can share these slides
Oh,
too.
yeah, absolutely.
Yeah, so these are other types of information you find on chemical labels. And the main types of contamination that you get or hazard is by inhaling it. So again, things that are very toxic, if you inhale them, you do in a chemical fume hood. It can be absorbed through the skin, or you can ingest it. So for this reason, like, never ever ever eat in the lab, don't bring liquids into the lab. It can really stink if you're doing a long experiment. You can't have your water bottle, but lab spaces are always or should be separated from where you eat and drink. Another big one is don't chew gum in the lab, and don't put on chapstick and do makeup, because there can be stuff or chemicals that are floating around and ends up on those things, and then you put it all over your face. So those are the main rules there, so don't do this. I love too that the person's sneaking it. So don't drink in the lab. Another big hazard is with sharps. Depending on the experiments you're doing, you might be using blades or potentially needles and syringes. And a huge rule with syringes, like the number one way people get accidentally injected with stuff, is when they try to recap the syringe to throw it away. So it will be in every health and safety quiz you ever take. Do not recap syringes. So once you take the needle off, the cap off, you throw away, when you're done with the needle, you throw it away into a sharps container, because it's when people try to do this that they end up sticking themselves and then unintentionally injecting themselves with something. So that's the main thing with sharps. Another thing to be aware of is a lot of labs are now replacing things that are sharp and breakable with plastic versions or safer versions. But a big one that I have broken multiple mercury thermometers, because sometimes I move too fast and can be a little clumsy. So that's a big one to put in the back of your head. If you break a thermometer and you're not sure if it's a mercury thermometer, that's something you want to contact your mentor or health and safety, and they'll clean that up properly. And the two big pieces of equipment that make you safe that you will see in almost all wet labs are a chemical fume hood and a biosafety cabinet. So the chemical fume hood actually has an exhaust system, and so it pulls out fumes. So again, things like alcohols, things that inhaling it would, like formaldehyde, you would want to inhale. You do in a chemical fume hood, like the more dangerous chemicals, things that are flammable. A biosafety cabinet doesn't have its own exhaust system. What a biosafety cabinet does, you work with cells, recombinant DNA material, tissues, things that you want to keep sterile. And a biosafety cabinet keeps air circulating, and it creates essentially a curtain of air between what's inside the cabinet and what's outside the cabinet. We'll go over this more when we do the cell module on the 2nd of June. But essentially you work with your hands inside of it, you don't move them in and out, and it keeps contaminants, bacteria, viruses inside and sterile, and it keeps outside air and contaminants away from that, and it keeps those things away from you. So those are the big differences. Dangerous chemicals go in the chemical hood, biological material in the biosafety cabinet, and the chemical hood has an exhaust system. So this is, we'll share these slides, but this is an example. And the other key things, and we'll point this out in the lab, these pieces of equipment, they are calibrated so that you keep this, it's called the sash, this glass door in front of it. And they're calibrated so when you turn them on, they're supposed to be at a certain height, and that that keeps the airflow properly and things, the fumes or the biological material from essentially escaping. So usually you'll hear alarms if you don't have it at the right height, and it's an annoying sound, so you adjust it where it should be. But that's another thing to be aware of, and we'll point that out as we use these equipments. So obviously, you don't want to fill your, when they're not in use, you keep these closed. You shouldn't be storing a lot of chemicals in there, especially flammable and explosible chemicals that can explode. This is just on like proper way to carry. You don't want to be carrying open containers. When you're transporting between labs or in public spaces, you want to use carts. You want to make sure they're capped. And again, a lab will have like standard operating procedures. So the nitty gritty of what you need to be aware of will be gone over in your internship. And then big things, especially in the summer, you need long pants, you need your toes covered. So today was raining, so hopefully nobody has sandals on. But definitely as we're working in June, I know it's hot, you can always change your shoes when you get here, but you don't want to spill or drop things on your feet and hurt them, and you want to keep your skin covered. A thing I always did in the summer when I want to wear shorts to work because it's 100 degrees is I would bring scrubs or a pair of pants to just throw on over when I was in the lab. So we're going to practice this when we're in the lab. We're always going to have on our safety glasses, our lab coats, and our gloves. We shouldn't be having any explosive chemicals. We do have some that are corrosive, but there's a lot of dangerous stuff in a lab. So it's better safe than sorry, and also the lab coat keeps your clothes clean. So when you're working with dyes and stuff and bleach, you're not getting that and wrecking your clothes. The goggles, I know Columbia's rule used to be whenever you're pouring liquids, you have to have goggles. For my company, you have to have them on whenever you step into a lab. So even people that deliver equipment have to put on goggles. But it is a good practice. Once you get something in your eye the first time, you will always wear your goggles. So I don't want you to get anything in your eyes. We will just wear our goggles. And just anyone, if you have glasses for what we're doing, I don't think we have the over the glasses goggles. You should be fine for what we're doing with your plain glasses. Different companies have different rules about that. But if you do want over the glasses goggles, we can get those for the few with glasses. We will share these slides. There's different types of gloves. Some gloves, when you're working, I've worked with more dangerous chemicals that can eat through plain gloves. So you have to wear more protective gloves. So it's just something to be aware of. We're going to use general gloves for what we're doing. We will go over this today, but there's a proper way to remove gloves so you don't get stuff all over you, and we'll go over that. You don't want to touch common surfaces with your gloves, even if you know they're clean. Other people don't. And if you've been touching the lab bench, you don't know if someone spilled something and didn't clean it up fully. And then you don't want to go touch door handles and elevator buttons. So generally, you don't want to be walking around in the hallways with your gloves. So if you're exiting the lab, you remove your gloves. For fire safety, the main thing I want to say, because this is something they check all the time at Columbia and it's important, label all of your liquids. If you have even a bottle that's distilled water, you want to make sure you label on the outside what that chemical is, and you can't use the chemical name. So for ethanol, you can't say ETOH, you write out ethanol. And the reason for this is if there's an emergency, well, there's two reasons. If there's an emergency and the fire department has to come in and they see all these bottles of clear liquid, they don't know if that's ethanol, which is highly flammable, or just water, and it makes the scene much more dangerous for them and chaotic. So you must label every time you make a liquid, basically, that you're going to store and cap with your initials, the date, and the name of the chemical inside. Water, you can write H2O, because every fireman will know that H2O is water. And then the other thing is, Again, labs have lots of people coming in and out, and things get dropped and come back to. So you'll see shelves and shelves of buffers and reagents that are made, and if you want what it is, your initials and the date, so again, people can know, can I use this buffer? Was it made 1 year ago? Was it made a week ago? So anything you're making and stacking up on a shelf, you always want those things for organization as well. And if I can't use it, I'm going to dispose of it. I need to know how I'm going to dispose of it. If it had ethanol in it, I can't put it down the sink. If it's just water and salts, I can put that down the sink. I will say these are just general, like, again, logic, but like don't put glass bottles on an edge, make sure things are stored securely and labeled. Don't put things in windowsills where they can maybe fall out a window. In these labs there are no windows because we're in the 3rd floor basement. Again, beware of where you store things. Flammable things have very specific ways, and explosive things where they can be stored. So this is a picture of a flammable item that was stored in a refrigerator that wasn't graded for flammable stuff. So instead of, when it exploded, instead of containing it like these special fridges do, it obviously didn't contain it. Yeah, so there's proper ways to store all of your chemicals. Acids, strong bases, flammables, oxidizers, all have a specific place they're stored. Your lab will have that information. So just beware, like all chemicals aren't in one place, basically. And in general, keep a tidy lab space. It keeps it from being a fire hazard, and also it helps you stay organized when you're doing your work. And then you'll see these in most labs, especially labs that have CO2 tanks for incubators. So compressed gas cylinders are used for different things. They always need to be strapped down. I will point out, if it hasn't been already, the eye washes in the safety shower and the fire extinguisher. Some labs will have trainings, like we always did a summer training where you got to set off a fire extinguisher and put out a fire. That's fun. Hopefully the BBL still does that, I don't know. But I will point these things out. It's just good to know, because in an emergency, like hopefully you never need these things, but you don't want to have to try to find it in an emergency. So we'll point out where these items are. Yep. You all know where the exit is, so as we go out of the lab, point it out, you came in the same way, that will be our emergency exit. And then spills, if it's a small spill, usually you can sop it up with like paper towels. If it's a chemical that is very dangerous, you would be doing it in the hood, and you would know ahead of time with dangerous chemicals what to do if it spills, because you want to know that before you use a dangerous chemical. If it's ever a big thing, like you drop a huge jug of ethanol and it breaks, that's when you can call in health and safety, or there will be somebody, it won't be a high school student, there'll be somebody that knows how to deal with those big spills. So if you spill something, I think the big thing here is it's okay, things break, things spill, don't try to hide it or don't try to deal with it because you don't want to tell your mentor or the person you're working with that you had an accident. It's better to be safe and dispose of and clean up things properly. Yeah, so then the last thing is things in a lab. Again, chemicals have to be disposed of in certain ways. So in general, acids, strong bases, flammable things, corrosive things, anything that's in a special cabinet storage does not go down the sink. So there'll be waste containers for those. We'll point those out. Things that have skulls and crossbones, or the dead fish picture, don't go down the sink. So you want to make sure, even if it's a tiny amount that you're collecting it, there'll be waste containers to collect these things. And all chemical waste containers will have to have a label, and usually they look something like this. And the 3 pieces of information you always need on these labels are the full name of what the chemical is. So again, not abbreviations. The percentage that you put in, so they have an idea of when they're disposing of it, how they have to properly handle it. And then there'll either be a check or you write in what the hazard is. But almost all labels are some format of this. So you need to identify the hazard, write out what's in there, and write the percentage. Those are the 3 pieces of information you always need when you make a waste container. And these, again, are on the labels of your chemicals. And if you're ever not sure, just ask somebody. And then, of course, make sure your containers are capped. A lot of this is logical. And then there, for most things, Columbia's rule is all gloves go in biohazard waste. Let me see if I have a thing. Sorry. Do I have, great. I'm going to go here. So anything that has touched cells or chemicals, so tubes, cell dishes, your gloves, even if your gloves you just put on and like wash the bench. For Columbia, all of that goes into a biohazard bag and that's disposed of properly. They're red bags. Every lab I've ever been in has a red bag in either a box or a bin where they have a biohazard waste. Anything that you think this might break the bag goes in a special sharps container. So blades, syringes, sharp pipettes, all go in the sharps. So we'll point that out. But the main thing to take away is you want to be aware of, like basically anything, any of the lab equipment you've used that's throwaway equipment, like one-use equipment, goes in some type of red container, either a sharps or a biohazard bag. And all institutions will have guides. Usually it's required to be posted somewhere in the lab, but if you're not sure, there's often a guide up with like how you throw away. So the big ones for us are going to be sharps, so pipette tips. We're not using syringes, but this is a serological pipette tip. The type 1 is a micro pipette tip. Will all go in our sharps containers. Gloves, tubes, dishes will all go into the biohazard. So, yeah.
Can I add one thing?
Yeah.
Sometimes it's tempting to, like, you say, oh, I put these gloves on, but I didn't touch anything. I can just throw it on the regular garbage. The reason you don't want to do that still is because if you think about the guy who's going to come get the garbage out on Monday morning,
Yeah.
he doesn't know what you used that glove for. Maybe you didn't do anything with it, or maybe you touched some cells and, got it contaminated. So it's just, think of it from that perspective. If you're like, you wouldn't know what this touched or was used for in the regular garbage bin, just be safe, put it in the red bag. That's sort of a catch-all.
Yeah, because like once you get in the lab and get comfortable, you know what a hazard is, and you're like, oh, I didn't touch. It was just PBS. This is fine. But somebody has to come take that trash out for us, and they don't know what we use that stuff for, or if they can trust us. So it's better as a habit just to put it. So I think that is it. Are there any questions about any of that stuff, or just from this morning?